Yeah, hi. Thank you all for staying with me. I'm full aware that I'm standing between you and conference dinner, so I'm trying to make this as exciting as possible. Also, some words of warning. This is a problem that I have and that I'm trying to think about. I don't have any solutions, but I am currently trying to address like how the biotic abiotic distinction, so the distinction between triggers of living things and triggers that do not live, how it operates in immunology. And I guess like this is followed by another question, which means what is this distinction between biota and abiotic? Is it the same in every discipline, or do different disciplines offer different kind of distinctions? You could also call the talk like in search of the biotic abiotic distinction in immunology, because I'm not sure how it is present, but I'm gonna try to hint at how I think it operates. So what I'm going to do first is contextualize this distinction a little bit and look like how origin of life research, evolutionary biology, and immunology try to distinguish between biotic and abiotic triggers and why this distinction matters. Then I will talk about small RNA-based immune responses in plants and invertebrates, because that's also the place where I'm coming from. So I did my PhD on small RNA inheritance, and I think small RNA-based immune phenomena, particularly not invertebrates, are an interesting case to reconsider the biotic-abiotic distinction we currently have operating. Then I will address whether there is a soft biotic-abiotic distinction in immunology, so meaning that while we do think that abiotic can cause immune reactions, there are still some qualitative differences immunologists make between living and not living triggers. And finally, I'm going to try to conclude, which means in my case, ask for questions. So I'm really excited to have you guys here also to pick onto your intuitions that you have about this distinction, because, yeah, it's something I've been thinking about like for a year, and I'm just telling you where I'm at right now. So what could the biotic-abiotic distinction be? I think I'm just starting with this intuition that these two things are different in important ways, right? I mean, if this is a laboratory mouse with a microscope, they also have things in common, right? Both techno-scientific objects that had to go through a lot of standardization procedures so we can like interact with them. But still, the fact that this thing is living and this thing doesn't is something important. Also, that I'm standing here and this table doesn't do the same things as ours is also like an important distinction. But we shouldn't base everything we think not on our intuitions, but also like think more conceptually about it. So what does origin of life research do with this distinction? So, I mean, obviously, you need a good working definition of what demarcates the living from the non-living if you're going to identify what was the origin of life. One way of addressing this is formulating necessary and sufficient criteria, and also historically, origin of life research, in particular, the concept of what the organism is very much connected to criteria for life. We have a lot of different approaches, so you can define life by how life functions, by metabolism, or by biochemical and physiological aspects. However, biota and abiotic also have many things in common, right? So we believe that both are somehow subjected to physical and chemical laws. And that's a whole debate about reductionism or anti-reductionism, meaning, can I explain everything that occurs in living systems in the end by referring to certain chemical and physical laws? That's not what I'm going to talk about, but it fits here. Usually it is thought that life adds a certain layer of complexity to things. Certain traits have been defined. I'm sure most of you might have gone through these in introductory biology lectures, but usually we think that characteristics of life are cellular organization, the ability to reproduce, growth and development, energy use, homeostasis, responsiveness to the environment, and the ability to adapt. The question is, of course, is this a characterization, or are these necessary and sufficient criteria? We have sort of like gray area cases, right? We debate, are virus living things, are transposons living things, but this is somewhat a good enough characterization that NASA, for example, would characterize something as living that they identified in outer space. So how are these things going in evolutionary biology? Another area that deals with living things and also the distinction between living and not living things. So Functionalism is here quite prominent, meaning something lives by the way it functions. And so living things can evolve emergent properties such as reproduction, metabolism, behavior, consciousness, and maybe also immune system, which is also often looked at as one of these big evolutionary transitions. And also like living things have the capacity to be even subjected to these major evolutionary transitions. For example, the transition from unicellularity to multicellularity. There is also, if we think about how evolutionary biology studies this, there is a difference between biotic and abiotic triggers, because we often talk about this kind of arms race between organism and the biotic environment. And this makes also particular innovations possible. So if we think about immune cells, like how certain mobile genetic material inserted into vertebrate DNA made complex immune cell functioning possible. Also, you can acquire parts of living triggers, right? For example, transposons. You cannot really do this with abiotic triggers. You cannot acquire a shift in temperature, for example. And also the interactions are a little bit different, because it's exactly what makes certain, say, bacteria living that our organism responds to, right? They are a threat because they reproduce. They are a threat because they metabolize, and like their metabolization products might be toxic. However, abiotic triggers are also quite important if we look at the history of life, right? If we look at the Great Oxygenation Event, which was probably the biggest mass extinction we ever had. There is also the climate crisis more recently, where we really care about our evolutionary trajectory potentially. And there is some terminological differences, I would say, between terms like resistance, tolerance, immunity. Immunity usually used to responses against the living. Resistance tolerance also used against about responses to the living, but also used for responses to abiotic triggers. So this is a bit messy, but that's kind of the terms we're using. Now, what's going on in immunology? How do we differentiate here? So if we look at textbooks or major reviews, abiotic triggers are not really considered, and there's also seems to be no need to distinguish between the living and not living. So that's not like something basic to start with, for example. To understand what an immune system is, what immunology studies, we of course also need theories of immunogenicity. So how the organism demarcates itself from the environment. And depending on immunogenicity theory, different components of what traditionally is considered the environment will be considered part of the organism. I think the best current cases, for example, the gut microbiota, like the holobiont, which means the host gut is microbes, because if there are certain traits, for example, expressed, think of the vampire bat, right? The vampire bat, it can metabolize blood, but it can only metabolize blood because it has gut microbiota, which make it metabolize the blood. So there's a certain trait that can only be there because host and microbes express it together. And since these microbiota are tolerated by the host, why not think about it as part of the organism, but used to be the environment? However, in current philosophy of immunology and also like new branches of immunology, such as ecoimmunology, there's usually a focus on the biotic parts of the environment. And this does not mean that they're ignored. There's just a bigger focus currently, for example, on host microbiota interactions. The distinction within the environment between biotic and abiotic triggers is more prominent if we talk, for example, about the units of selection, meaning which are the structures that natural selection can act upon. For example, we have in the niche construction debate the question, is H2O actually a part of the unit of the selection, because it is sort of required in the network of structures that maintains the stability of traits. So usually people that do not think this is the case point to H2O not affecting differential fitness, right? Because what belongs to everyone belongs to no one. And on the other hand, there's also no active maintenance, meaning there is no reciprocal interactions between H2O and, say, an organism. So the environment for immunology, usually we consider most the living parts. In other disciplines, we have like a more living debate about what to include and what not to include. So what are intuitions about the biotic-abiotic distinction in immunology? One thing is that one thing that's very important about immunogenicity theories is that it's very often about specific interactions. And very traditionally, we have already heard today that like the distinction between adaptive and innate immune system is not very adequate anymore. But adaptive immune system have been this paradigm example of responding specifically and being capable of granting specific immune memory, meaning a specific memory against one particular trigger. This is also this distinction between reactivity versus specificity, right? The choice whether to respond to a broad range of triggers or only a very small range, but very effectively and specifically. For immune recognition, we usually think of an epitope being required, right? And this would be like a molecular structure that is specific to a particular trigger. Epitopes are presented by antigen-presenting cells together with second stimuli to T and B lymphocytes who recognize the molecular unique epitope. And this can trigger an immune response. This is hard to think about when we consider abiotic triggers, right? Because a lot of abiotic triggers do not have a body, meaning we cannot really think about the epitope in the terms we usually think about them because they're not corporeal, meaning they're not entities and they're also not derived from entities. And thus, there cannot be like this kind of direct receptor epitope interactions we usually think about when we think about immune interactions, because there is no immune receptor for sensing it's too cold, it's too hot, there is too much salt. Are there adaptive immune responses? Are there specific immune interaction against non-corporeal abiotic triggers? Because immune cells must respond to abiotic triggers, right? We know that alum in vaccine is an adjuvant that increases immune reactions. We know that fever is something that's really important for the immune system. There are dead bacteria in the body the immune system needs to interact with, and also our body temperature just changes within 10 degrees from head to toe, right? So immune cells need to be somehow responsive to these abiotic triggers. And there are quite some examples of work on responses to abiotic in the immune system, right? There is, for example, work on temperature sensing by T cells. There's work on how osmolarity changes induce T cell inflammatory responses. And there's also work on how the nutritional status affects immune cell metabolism. The thing is, however, we do not have really a conceptualization how there could, for example, be immune memory of a particular abiotic trigger. However, so far, we have looked only at vertebrate immune systems. And I think a good question to ask is this distinction between biotic and abiotic is somehow shaky and not very well expressed if you look at theorizing or practices. So maybe if we would look at different model organisms, we could learn something more about this distinction and how it operates. So I'm going to go into small RNA-based responses in plants and invertebrates. So just very short reminder of what's going on in plants and worms. So plant immunity, if you will read any kind of review on what plant immune responses are, they are usually characterized by a lack of, right? They lack dedicated immune cells. They seem to lack adaptive immunity. Usually plant immunity is characterized as those responses to viruses, pro and eukaryotes, and we have like a whole lot of different immune pathways. We have PAM-triggered immunity, we have antiviral receptor effector triggered immunity, we have cytokines, we have volatiles, and we have RNA interference. Similar things are going on if we look into how C. elegans immunity is usually written about. Again, we have like this characterization of C. elegans lacking a lot of stuff, right? It also lacks dedicated immune cells. It would be very remarkable if C. elegans had immune cells, right? Because it's only like 1,000 cells in total. So if it only would have like 15 immune cells, it would be a huge lot of the whole C. elegans. Also, it is usually said not to have adaptive immune system. Also here, when you look in literature, you usually conceptualize responses to viruses, pro and eukaryotes as those things that C. elegans has an immune response to. And here again, we have a lot of different immune factors. We have MAP kinase signaling, insulin signaling, tumor growth factor beta, antimicrobial peptides, lysozymes, reactive oxygen species, and also RNA interference. And we're going to focus on this. And I mean, RNA interference might also play some role in vertebrates. I'm not too much of an expert there. I think I read a lot of papers that say, well, these RNA interference pathways, if they infect mice, for example, influenza, they start like 2 weeks after infection, which is like not too good for the mouse's survival. And like small RNA-based responses alone wouldn't like do it for vertebrates. So they are much more prominent in these systems. Quick reminder, what are small RNAs? So in principle, they are short molecules that complementarily bind to usually messenger RNA, and the canonical result of this binding is the degradation of the messenger RNA, and that's why they are usually attributed to have a big role in gene silencing. They are phylogenetically conserved, meaning most species have them. Those that don't have them anymore, I usually believe they've lost them over time. There are many different small RNA species, and there is a multiplicity of physiological functions. That the terminology is a mess, right? If you have a microRNA in plants, microRNAs do a lot of different things in humans, so you can't really infer the functioning by the people. There's also other really cool things about small RNAs, and this is very specific to the cases I'm talking about. Small RNAs are in principle amplifiable, not in humans, but there are a lot of species that have RNA-dependent RNA polymerases, meaning similar to how there is DNA-dependent DNA polymerases. RNAs can also be amplified. They are responsive to endogenous and exogenous biotic and abiotic triggers, so they're sort of like these master regulators within the cell. And what's also really cool, and which a thing I was very excited about during my PhD, is that their effects can persist transgenerationally. So they might actually also be a cool bridge between for the transgenerationality of the immune system. But I think what has been phenomenologically at least quite well established in C. elegans is that if there are certain triggers that find the organism, for example, virus attacks, or there's a heat shock, or nutrition is just very short, there are certain specifically responsive small RNAs that on the one hand can be amplified, that have a lot of crosstalk with histone modifications, in the case of plant or the DNA modifications. And through these amplificatory cycles, they can persist throughout several generations. So you can see that, for example, the third generation of an infected worm still has, for example, small RNAs that are complementary to a particular virus, or that still can address food deprivation. Similar things have also been shown for plant model organisms. Particularly, a lot of stuff has been shown in Arabidopsis. So why consider small RNAs immune effectors even? I mean, descriptively, if you look at that part of immunological literature, they are considered immune effectors. But I don't want to presuppose what is just descriptively here, but also motivate my case why I do think they are really cool and important immune effectors. So on the one hand, they are responsive to stresses and pathogens. They launch specific responses with regards to these stresses because they have this kind of complementarity-based functioning. They also interact systemically with other immune effectors. So if you have a systemic perspective on what all the interactions are that comprise immune system functioning, we also have small RNAs here as important players. And also they persist and instantiate immune memory. And this, of course, has a lot to do with half-life, but also there are also a lot of cases where they particularly also amplified in order to persist longer and guarantee this kind of immune memory for a longer time. So let's go into some examples. If you look at Arabidopsis thaliana, small RNAs have described to launch a whole host of different responses specific to biotic and abiotic stimuli. We know there are small RNA-based responses to fungi, viruses, bacteria, heat, cold, salinity, or drought. There are, in principle, two ways how small RNAs can act. Either they target stresses directly, for example, because most viruses or all viruses have an RNA phase. There are small RNAs that can directly target viral RNAs. However, they can also induce other specific gene regulatory changes. For example, after heat shock, they can affect heat responses or messenger RNAs that are involved in the heat response in the cell. So just to give an example, there is just a whole lot of different small RNA pathways, both when we talk about biotic triggers, here bacteria and viruses, but also helminths and fungi. And a similar thing is the case when we talk about abiotic triggers such as drought, salinity, cold, heavy metals, temperature, nutrients, hypoxia, and UVB radiation. And also very importantly, there is a lot of crosstalk. There's also a lot of crosstalk when we look mechanistically at small RNAs addressing biotic and abiotic triggers. One particular example, I think because it was quite famous, is RNA-based immunity against viruses. It has been shown that antiviral RNAs persist after infection for several days and weeks. And this has also been adapted as a technology to immunize plants against viruses. Another case for an abiotic example, small RNAs in heat stress memory and resistance, there's the microRNA 156. It is necessary and its overexpression is sufficient for heat stress memory. It operates through a gene regulatory cascade involving many developmental regulators, and it induces increased tolerance to further exposure. So this lasts quite some time. This also happens in C. elegans, and there's a very important caveat relating to the generation times, right? C. elegans lives for 3 to 5 days. So usually when people measure how small RNAs can guarantee resistance or immunity to a trigger, they look in the next generation because it just also makes like experimentally more sense. But here it has been shown, for example, that resistance to virus can be guaranteed through small RNAs through their complementarity. But also resistance to starvation can be guaranteed by small RNAs that are specifically complementary to messenger RNAs involved in log metabolism. And both these phenomena have been shown to be also heritable. Both pathways emerge at the level of effector proteins, meaning that both the abiotic and the biotic small RNA responses merge at the level where they compete for particular effector molecules, such as argonauts, disers, RNA-dependent RNA polymerases, and so on. So if we would just look at Arabidopsis and C. elegans stress responses, and if we agree that small RNAs are somewhat immune effectors, the distinction between biotic and abiotic stresses does not seem intrinsically straightforward, because biotic and abiotic stress pathways mechanistically converge. So if we are prepared that it makes sense to say that small RNAs are immune effectors, why differentiate from those that are induced by abiotic stresses? And if you look into the immunological literature on C. elegans and Arabidopsis, you will not find any documentation of small RNAs against abiotic triggers. So somehow there seems to be a distinction intact. If this literature tries or covers abiotic triggers, they are most considered influences, for example, the influence of temperature on disease susceptibility or fine-tuners of immune effectors. So it seems that there's this kind of soft biotic-abiotic distinction. Abiotic triggers are not believed to not affect the immune system at all, but it's just not believed that specific interactions with the immune system and specific memory to these triggers is possible. And this seems somewhat similar to our short vertebrate case, right? Because also before I said, well, they are considered fine-tuners. They are considered to have effects, but we're not considering them as they're, for example, being a B or a T cell that can guarantee after like re-exposure particular immunity against the trigger. So why is that soft biotic-abiotic distinction there? And I think one explanation is the dissimilar situatedness of the small RNA case, which is that of epigenetics versus immunology. So if we look at the history of small RNAs and small RNA biology, they are very much embedded in the language of biochemistry and genetics. And a lot of the phenomena that are considered now immune phenomena of small RNAs in C. elegans, in plants, have also been established in biochemistry or genetics lab in the late 90s and early 2000s. And it's very interesting, and particularly when we look at the epigenetic research on this, the soft distinction is not operative, and there's a holistic notion of the environment. And this could maybe be even said for epigenetics in more general, right? Because usually we talk about certain environmental triggers, be them biotic or abiotic, usually in one wash. And that's particularly the case if you look at small RNA inheritance in C. elegans. So it seems that on the one hand, like from the discipline small RNAs were born, there seems to be not an operative biotic abiotic distinction, but then small RNAs were imported into immunology, and there suddenly this distinction is operative. So one reason could be that there is some disciplinary imperialism of vertebrate immunology, which is fair enough, right? Because immunology traditionally just is a discipline that cares very much about understanding, curing, treating human diseases. And so also I think by sheer numbers, we just have like most immunological work still done on chilled vertebrates model organisms. And that's also why plant and invertebrate immune systems usually don't make it into textbooks, at least not the ones I found. There is very often a focus on homologous effectors. So those things that are similar in the worm, in the plant, are usually better understood and more researched than the things that are dissimilar to chilled vertebrate immune systems, right? Because obviously when something's similar, it's more clear to count it also as part of an immune system than something that is very dissimilar. So it seems that immune phenomena are to an extent appropriate in a discourse shaped by the properties of chilled vertebrate immune systems. So as I already said before, there's this particular notion of specificity we might attach to immune recognition of T and T receptors. We have this problem of the non-corporeality of particular abiotic triggers. So how could there be an epitope? How could there be specific immune memory against them? And these things also in chilled vertebrates are studied in epigenetics, where very often the abiotic distinction is not operational, but they're usually not studied in immunology, and we have a soft biotic abiotic distinction operational. This I think I already said. So I think one thing that happens, and happens particularly for the small RNA example, is that there is a certain clash of representational spheres. Because if you usually look at basic research, we pick these organisms we think that are simpler, and they're supposed to represent all kinds of organisms. However, when we have the applied research context, we have particular model organisms such as the mouse that defines the range of processes in the applied research. And I think to a degree, what happened to C. elegans immunology is that sort of like the representational sphere of the mouse model organism was imposed onto the whole range of phenomena that could be immune phenomena, and those that are similar or somewhat similar to the mouse model organism have primarily been focused on also in this part of invertebrate immunology. So how could we take that further? Where are we at? If the soft biotic abiotic distinction is descriptively accurate for immunological theorizing, so if it's somehow there, what is the normative import? So do we like that it's there? Shouldn't it be there? What to do with it? I think one question we could ask is, should it be less universally applied? So I think there seem to be good reasons maybe why a soft biotic abiotic distinction makes a lot of sense in vertebrate immunology. However, if there is a certain multiple realizability of immune systems, as we have seen, for example, for CRISPR-Cas9 based system, or that there's a lot of immune memory also in non-chilled vertebrate organisms, maybe one distinction just does not fit all. And I think that only investigating less studied model organisms can help us to understand the generality of our assumptions, or where the limits of our assumptions are. Also, we could ask the question, do we need a different concept of the immunologically relevant environment? So if we conceptualize the environment, which factors do we think are important? Obviously, we always make choices when we think about the factors we study, but we can think about the justifiedness of these choices. So far, as I said before, there has been this focus on the biotic environment. However, if we consider that there can be specific interactions of immune effectors with abiotic triggers, does this actually affect also immunogenicity theories, or the question of what is part of the organism? This would be super unintuitive. However, I think we could rethink this, for the cases where there are specific interactions with abiotic triggers. Lastly, maybe Also, the small RNA case also points towards a different theory of immune cognition, right? Because the idea of the small RNA state, meaning not that a single molecule, but always like a quantitative amount of molecules can represent the environmental trigger. And the small RNA state presenting a current status of the system might focus like a presentational model of immune cognition over the representational model. And this meaning that the cognized and cognized distinction is sort of like dissoluted. And lastly, I think at least small RNAs emphasize the organism's constant interaction with an embeddedness in the environment and the environment in its totality. And it can show that that non-corporeal environmental abiotic can specifically can be specific interaction partners. And yeah, I'm super excited to hear what you think. I've been like struggling with this distinction for quite a while, and yeah, excited for your question.
So there is ample room for questions. So yes, please, Bob.
So yes, thank you very much for the presentation and for the provoking talk. And so, if I understand you correctly, as because there are many threads like that that you raised, but one problem is this: that if, for example, we have worms, right, and their immunity is mediated by a small interfering RNA, right? Then this small RNA can mediate sort of defense or protection against not only bacteria, viruses, and so on, but also against what you call abiotic triggers, like, for example, hunger, I don't know, some temperature changes, and so on. So we have the same mechanism, mediating protection against bacteria, viruses, and also like environmental factors of different kinds. And now you're wondering, like, why isn't it the same in immunology, invertebrate immunology, right? More or less. This is how I understood it. And here we have a focus on a particular kind of triggers, like bacteria, viruses, and like corporal triggers, as you mentioned. Maybe some organic molecules of different kinds, right? That may also trigger an immune response. Then also we may have a sterile inflammation. There is no microbe involved, no corporal trigger in a sense, but we have an inflammatory response in sterile conditions. So now we are wondering, like, why, if, for example, in the worms we have the overlap between stress responses and immune responses, why don't we see the same in the vertebrate immune system? But here, my worry is maybe because there are just different pathways involved. That we also protect ourselves from temperature changes, hunger, and so on. And now this is mediated by different, not immune cells, and maybe, I don't know, maybe we are hinting that we would like to expand the notion of immunity also to incorporate these kinds of responses. Do I understand it correctly? And maybe, this expansion of the notion of the immune system would be kind of beneficial, would open some new perspective for understanding the relationship between stress responses and immune responses. Do I get it?
So I would just only partially say that I want to expand into also about the triggers in general. My point of departure is more that looking at the small RNA-based system in C. elegans, if you would know nothing about vertebrate immunology, would have like one particular system that is launched again biota and abiotic of this mechanistic convergence. It seem pretty much to be like one system. However, if you read the literature in invertebrate immunology, on C. elegans immunology, you only find small RNAs that specifically interact with biota to be considered part of the C. elegans immune response. And my starting point is, why in this example do we still have this distinction operative when it doesn't seem just from what's mechanistically there necessary to have it? So I'm maybe for limiting the distinction, but not for giving it up, but just to look at the empirical matter of the particular organism and to be open that it might be different for different organisms.
Yeah, Judith.
I think I am struggling a little bit because if I understand you correctly, the small RNAs in CL, they are still fine tuners, right? They're not directly, they're sensing whether it be salt or heat, they're fine. And to me, the mammalian immune system has all of that, right? We detect hypoxia, we detect salt, we detect temperature, we have receptors. Mechanosensors is a really good one for abiotic. I mean, we sense how stiff, so we have those responses, and sometimes downstream of that is small RNAs, and sometimes it's a whole different pattern. But I guess I'm struggling a little to see that. I mean, of course, I totally agree that we should be, that when we look, we're too anthropomorphic. We look at, ourselves and then think secondarily about plants. But I think there's a lot of plant immunologists who come from a completely different direction, who think about plants first and then mammalian immune cells second, right? So I'm struggling to see what the real distinction is between what you describe and what we do know about the immune system already. Am I being clear?
So where I'm coming from is that this distinction seems to operate in my subject of study, which is invertebrate immunology.
Right.
And also if you look at the plant literature, even though obviously if you talk with plant immunologists, they care more about plants than about humans. However, if you read the reviews, you still find these things, right? They are described by a lack of. And there's a lot of emphasis on the things that are homologous to mammalian immune system function. So this is still there. And I'm not saying that it would be equally bad to now say just because this is going on in these model organisms, we should also import it to the short vertebrate context, because there might be very good reasons why this distinction beats off there is in place, and that the immune system just evolved to be a thing that mainly deals with biotic triggers, and there are others other interaction partners for abiotic triggers that are studied, for example, in toxicology and stuff like that. The question is just whether there is still the bearing of how this is realized in our most paradigmatic model organism that leads to us not being able to consider different instantiation for what the data shows us, but to make these exclusions based on how the immune system of short vertebrates work. Because I'm looking for an explanation or something. Why is this distinction made in the invertebrate context?
Is it not just historical?
Yes. So do I understand correctly then when you mean abiotic, you mean also non-material, let's say, because dead bacteria is abiotic or not?
And that bacterium wouldn't live anymore. It would be abiotic, but this would be corporeal. It would be a body.
But what you mean in the whole discussion is non-corporeal. Is that correct? Because dead bacteria is dead, but still we recognize it using precisely the same methods. Because some colleagues in immunology, which were defining via PEMS, for example, that were making the point that we do recognize differently live or dead bacteria, for example, and a lot of it would be based on RNA recognition, because RNA after death is very quickly degradable and so on. So we do recognize differently, let's say, a live and a dead microorganism. I'm not so sure that that is the case, to be very honest, but there were. But you're talking mostly between non-corporeal. Okay, but and I also was thinking during your discussion, because, for example, we have cold receptors, which are the same receptors for some certain chemical substances from hot peppers, for example. I forgot what's the name. Exactly. It's the same receptor basically to sense a chemical substance and to sense cold, for example. And there are many examples. So at a certain moment, it's a continuum. I think it's very, very tough to draw a line where are things moving from one to the other.
Would you still agree that you as an immunologist don't think about a response to a non-corporeal abiotic trigger as something you could have immune memory
against.
I would not know that.
Why would you need immune memory? I mean, we evolved.
I can give you an example of that. I don't know, is a lesion, for example, corporeal or not? So you make a cut. Well, it's the absence of something. And for that, Elaine Fuchs has shown memory, the memory of the cut, basically.
Yeah, I mean, that's something that's interested me a lot is why do we need memory for tissue repair, which we do have.
We do have memory.
But it may be that evolutionarily, so I'm of course interested in things like worms that bite your gut wall, right? They bite your gut wall and they suck your blood. And I think you do need to remember to repair that wound really quickly.
I would see more like a learning process than like a memory.
Right. But I think but it does, in that case, it does, I think, evolve antigen-specific T cells.
Yes.
So I guess the question is, what would be an abiotic thing that would be of value to have antigen-specific memory?
Well, that's the question, should be antigen-specific or not? If we think of memory in general.
Yeah.
I mean, if we live at the North Pole, for example, I don't know, and every 2 months we have frozen skin and fingers, it is actually useful to repair it quickly and better next time. It's basically a process. It's a fundamental learning process. If you need to do a thing 200 times during your lifetime, the 50th time you do it better than the first time. So it's, I would guess.
You submit, or because there is Jan has a question.
Sorry, yeah.
I would like to understand what you still think as the term abiotic to scan the field. What about the crystals? Presence of a crystal in organism could be detrimental, could be highly immunogenic, and we have the sensing mechanism how to get rid some crystals like silica, like urate, like cholesterol. Is there a part of the life for the other one? Because that's what I'm trying to bring to the scene is kind of like what Matt Singer wrote in Immunology. So the danger signal, sending of danger, and change in how we think about the immune system from the policeman to the fireman. So not patrolling all the time, but reacting only when there is a real need for reaction. So for me, the definition of this immune system will be a mechanism how to look for the danger and maintain the homeostasis of the organism in a very general sense. And identifying the crystals, obviously something non-living would be part of immune response. And there are the scavenger receptors, which are very potent to interact with and being part of endocytosis by the macrophages. And I think that part of reasoning about the limits of immunology is now becoming very popular with all these nanotechnologies and nanocrystals and nanobodies, which are linked to transfer of therapeutics or even providing the vaccine and over vaccine technologies. So I think it's not a trivial question, but it's about the body, something physically existing.
Exactly.
Is something like
we have a massive system, NLRP3 inflammasomes to detect the crystals. It's an industry. I guess the question I think I hear you're asking is, would it be useful for us as mammalian immunologists to think in abiotic and biotic terms? I think we do already. We just had to put it in that space. And I'm not sure what the answer, for me, I'm not sure that I have so many other concepts to deal with.
I mean, just to what you said, like a crystal would also be a body, right? So it would be corporeal. And I think like another example would be allergies, right? Because you can be allergic against a particular metal. which would also be clearly not living, but it's also a body. And I think there are specific interactions, but these are like secondary, like association with certain haptens, and then the immune cells can specifically interact, if I'm correct on that.
I'm confused because crystals are, I mean, the crystals that do things like cholesterol crystals, they're tiny molecules. They just form crystals, and then they trigger these responses. So maybe I'm confused by corporeal versus non-corporeal.
So for me, like corporeal is anything, like non-corporeal is anything that is not a body and it's not derived from a body, such as, for example, temperature, heat, something like salinity, for example. So a little bit like a distance, right? It's material because it exists, but it's not derived from a body, and it isn't a body.
Maybe one more comment to that. I tried to define very generally the immunology as the fireman which is going to the sea when there is a danger. So in your trial to us to accept that broader picture, I think it's relatively easy. We can add to what we know from the immunology, the feature protein biochemistry and responses, and it will be sensing danger triggered sensory mechanism. And I'm very open to accept these molecules to the immunology in a broad sense, because in reality, it's a sensing system. It's triggered by the danger, and it's reflecting or pointing to the homeostasis. So that's for me acceptable.
But it's a body. HSP are body in your definition, so it cannot do. I mean,
it's not body. It's sensing the osmolarity, temperature, and so on and so on. So,
but then HSP wouldn't be that specific, right? So the next question is, would it be because there are interactions with HSP and the immune system? Could we talk of specific immune memory regarding a temperature change? Is the immune system capable of immunizing against temperature, which isn't usually like two words that are used?
Oh, but we do immunize against temperature. UCP1, right? That's immune mediated, and we turn on heat. We do that as part of our immune response. Do we not? That's what we do when we're cold. And we do it as with immune memory. So it is fascinating. I think that because type 2 immunity drives thermoregulation, and I think it drives it as part of an evolutionary response that when we're faced with a cold winter, lack of nutrition, and high helminth worm burdens, we need to bring our body temperatures up. So it is there in the system.
But I would then more resistant to the next cold winter.
Yeah, I think the idea is if you are an and these are like they do all this work in the SOE sheep, but if you have a stronger type 2 immune response, yes. Yes, there is a memory.
and also the other question is, do you need that? I think it was a very good question. I mean, we can be convinced by Mihai and others that immunological memory is much bigger than we used to think. But do we need immunological memory in our definition of immunity? You don't have it in the danger definition, for example, which I don't think is valid, but this is another debate. So all the definitions on the market, I think, now do not consider memory as a necessary component. So this is something that you would probably have to clarify. Are you talking about the need for some memory in immunity, and then the consequence would be, what about even memory to non-corporeal processes or something like that, or are you talking about immunity more generally without the need for a focus on memory? So that is something that I think was not entirely.
Well, and I think it makes me think it's like, the fingers, freezing in the winter. What happens is you change your extracellular matrix so that the next time it's not necessarily immune memory, but you literally physically change so that you're more resistant in a physical way to some of these things. So then that becomes, not that it can't be immune controlled, but
Could you suggest could you suggest some, for example, some experiments or some questions that experimental immunologists could try to address that they don't, a little bit in the spirit of what we are saying here. Yeah. So what is a suggestion that, if you have as much money you want, you just say, do that. What would that be, given your suggestion?
Right. And obviously, as I'm a small RNA person, I would be super interested in, I think there are a lot of stress responses in short vertebrates that where small RNAs are also triggered. And right, as there was at the time a transition between a more small RNA-based system that also handled a lot of immunological concerns for organism, and then the short vertebrate immune system suddenly coming to stage, it would make sense whether there would be a lot of interactions. So for me, it would, for example, be very interesting how small RNA-based stress responses in short vertebrates interact with immune responses to maybe reciprocally guarantee some sort of resistances, immune memory to these kind of triggers.
But is it all just about small RNAs? Because I think you're conceptually, it isn't just, because we probably respond to these, we certainly as mammals use small RNAs, obviously, but we probably have all sorts of other mechanisms, many of which we probably haven't even worked out yet.
It's not about small RNAs. It's just my area of expertise, right. And where I was coming from my invertebrate examples.
Yeah.
Where you have this system that addresses both triggers pretty much the same way, but if you talk about it immunologically, it's only one part, which is the biotic part, being considered as part of this kind of immune system response. And it's also, I think, an interesting case where the epigenetic and the immunological could very nicely overlap, because you have small RNAs on the one hand as these new hot candidates for transgenerational inheritance on the one hand, and where the immunological adaptiveness and the evolutionary adaptiveness meet this, so it could be just a very cool target for this.
I think the transgenerational stuff would be really fascinating for things like cold adaptation. Whether that's immune mediated or not, but we all know that a child that grows up in a very cold environment is better at handling cold than, but that's whether that then goes on to the next generation. I don't know.
It's interesting.
Yeah.
So I don't see any questions here. I don't see any questions on Zoom. So unless someone is still eager to speak up, no. All right. So then let's thank Sophie